Welcome back. In Module 1, we laid the biological and physiological groundwork for industrial biochemistry, discussing microbial physiology, genetics, metabolic pathways, and the principles of continuous culture systems. In Module 2, we move from understanding the organisms to engineering them and their environment for maximum productivity. This is where industrial biochemistry meets industrial engineering. We will discuss strain improvement, the methods by which microorganisms are enhanced for higher yield, better stability, and tolerance to industrial conditions. We will examine fermenter, bioreactor design, because microbes can only be as productive as the vessels and conditions we provide. We will cover downstream processing, the crucial stage where we recover and purify the product from the culture. By the end of this module, you should be able to appreciate how strain selection and modification, bioreactor optimization, and product recovery are interlinked in creating an efficient and profitable industrial process. 1. Strain improvement in industrial biochemistry. The productivity of a fermentation process largely depends on the capabilities of the microbial strain used. Even if a wild-type strain produces a desired product, yields are often too low for commercial viability. Therefore, strain improvement is essential. 1.1 Objectives of strain improvement. Higher product yield, increase the amount of target product per unit substrate. Faster production rate, reduce fermentation time. Tolerance to inhibitors, resistance to product toxicity or byproducts. Utilization of cheaper raw materials, ability to ferment unconventional feedstocks. Genetic stability, retain desired traits over many generations. 1.2 Traditional method of strain improvement. Before recombinant DNA technology, strain improvement relied on natural variation and selective pressure. A. Mutation and selection. Random mutation can be induced using chemical mutagens, e.g., nitrosaguanidine, EMS, or physical mutagens, e.g., UV light, gamma rays. Mutants are screened for desired traits such as increased enzyme activity, resistance to feedback inhibition, or improved tolerance to high product concentrations. Example: mutants of Corynebacterium glutamicum that overproduce lysine due to deregulated feedback inhibition in the aspartate pathway. B. Adaptive laboratory evolution, ALE. Microbes are grown under selective conditions for many generations. Gradually, strains evolve traits like substrate tolerance, temperature resistance, or altered metabolic pathways. C. Protoplast fusion. Involves removing the cell wall of two different strains, creating protoplasts, and fusing them. Allows genetic recombination between strains that might not naturally exchange DNA. Example: hybrid yeast strains for improved brewing characteristics. 1.3 Modern method of strain improvement. With genetic engineering, we can directly manipulate the genome to achieve precise improvements. A. Recombinant DNA technology. Introduce genes encoding enzymes for desired pathways. Knock out genes responsible for byproduct formation. Example: engineering E. coli to produce human insulin. B. Metabolic engineering. Systematic modification of metabolic networks to redirect flux toward desired products. Can involve overexpressing certain enzymes, deleting competing pathways, or altering cofactor balances. C. CRISPR-Cas genome editing. Highly precise tool for gene insertion, deletion, or regulation. Allows rapid prototyping of improved strains. D. Synthetic biology approaches. Designing and constructing entirely new biological parts and systems. Example: building synthetic operons to produce non-native compounds like artemisinin precursors. 2. Fermenter, bioreactor design and operation. Microbes require controlled conditions to perform optimally at industrial scale. A fermenter, or bioreactor, is designed to provide the right environment, including temperature, pH, aeration, and mixing, while preventing contamination. 2.1 Basic requirement of an industrial fermenter. Aseptic operation, prevent contamination over long production runs. Adequate mixing, ensure uniform nutrient and oxygen distribution. Efficient aeration, for aerobic processes, oxygen transfer is critical. Control systems, for pH, temperature, foam, and dissolved oxygen. Scalability, performance at large scale should match laboratory or pilot scale. 2.2 Types of fermenters. A. Stirred tank bioreactors, STRs. Most common type. Agitators mix contents, spargers provide aeration. Suitable for both aerobic and anaerobic fermentations. B. Airlift fermenters. Rely on air circulation rather than mechanical agitation. Lower shear stress, ideal for shear-sensitive cells. C. Packed bed and fluidized bed reactors. Used for immobilized cells or enzymes. Substrate flows through packed or suspended biocatalyst particles. D. Photobioreactors. Designed for light-dependent organisms like algae. Ensure uniform light distribution and CO2 supply. 2.3 Key design parameters. Volumetric oxygen transfer coefficient, KLA, determines aeration efficiency. Mixing time, how quickly conditions become uniform after changes. Shear stress, must be minimized for fragile cells. Foam control, mechanical foam breakers or anti-foam agents. 3. Downstream processing, DSP. Once the fermentation is complete, the product must be recovered and purified. This is often the most expensive part of the process, in some cases, 50 to 80% of total production cost. 3.1 Steps in downstream processing. Cell separation. Centrifugation, filtration, or sedimentation to remove cells from the broth. Cell disruption, if product is intracellular. Mechanical, bead mills, high-pressure homogenizers. Non-mechanical, detergents, enzymatic lysis. Product isolation. Precipitation, solvent extraction, adsorption. Product purification. Chromatography, crystallization, ultrafiltration. Product polishing and formulation. Drying, stabilization, blending with excipients. 3.2 Examples of DSP in industry. Ethanol production, simple, distillation after fermentation. Antibiotics, complex, involve solvent extraction, multiple chromatographic steps. Enzymes, purified via filtration, precipitation, and chromatographic polishing. 4. Integrating upstream and downstream operations. An efficient industrial process is holistic. Upstream strain improvement and bioreactor design must be in sync with downstream recovery strategies. For example, if a strain is engineered to secrete a product, downstream processing is greatly simplified. If the product is unstable, the fermenter must be designed to allow rapid recovery after production. Summary of Module 2. We've now explored the engineering and operational side of industrial biochemistry. Strain improvement from classical mutation to CRISPR editing. Fermenter design, choosing and optimizing the right bioreactor. Downstream processing, turning microbial products into pure, usable commodities.